Bismillah ar-Rahman Rahim. We start the last chapter, which is inheritance, and this is the video 3, which is continuation of the two videos which have been on this last chapter, which is inheritance. Now, in this chapter, we are just going to be discussing genetic engineering, and genetic engineering is a topic which is a very interesting topic, which is going to cover about how people as genetic engineers have altered the bacteria, and the bacteria now are producing a useful molecule for us which we need to treat certain diseases. Now, in genetic engineering, what we do is we take a gene that controls the production of human insulin and we insert it into a bacteria, and then the bacteria are cultured and the insulin is produced, and then the insulin is extracted, the bacteria is separated, and the insulin is removed, and then we give people injections of insulin, and we sell that as small bottles in which this liquid which contains the insulin is kept. Now, people suffering from diabetes need injections from insulin. Why? Because you see, their pancreas does not produce insulin. You remember there's the islets of Langerhans, and in that there are special cells which produce insulin, which are called the beta cells. Now, previously in the years before genetic engineering, we used to use animal insulin. So we had to slaughter an animal, then we have to take the pancreas out, and then we had to make an extract from the pancreas, and we used to get that animal insulin. We also used pig's insulin, we also used cow's insulin. So a lot of animals were slaughtered and a lot of animal organs were used. But there were problems with this thing. Problems were that people had a lot of allergic reactions, like some people are allergic to eggs, some people, like I'm allergic to mangoes. So people had allergic reactions to this animal insulin. Then there were a lot of religious issues, there were a lot of ethical issues. Religious means, like for instance, Hindus didn't used to want to use cow's insulin, as Muslims didn't want to use the pig insulin. And then ethical issues is that we were killing so many animals. And then, of course, another thing which was the most important thing is that in certain animal products, some diseases were being transmitted, like mad cow disease. We didn't know. It was caused by a prion protein, and we didn't really know this. So whenever we used animal products and we were giving it to humans, we actually were spreading diseases. So that was one of the biggest dilemmas. And in order to solve this problem, in came the genetic engineers, and they said, okay, let's transform bacteria and put in it the gene for insulin, and the bacteria will make insulin for us. Now, what are the basic tools of genetic engineers, and what are the basic steps or the events that we do? Number one, we take any human cell, whether it's anybody's, it doesn't matter, and we look on inside the nucleus, and in the nucleus, we've got the chromosomes. So chromosomes now somehow we locate and find out that there is a gene for insulin on a special pair of chromosomes, maybe the 7th pair or the 10th pair or the 11th pair, where we get this gene for insulin. So we take this out, and we know that this is the place where the gene for insulin is present. Now what we need is we need a scissor. Now the scissor is restriction enzyme. That's a DNA scissor, we call that. And I'll just tell you about restriction enzymes in a while. So restriction enzymes are obtained from actually bacteria, and we cut this gene for insulin. And we get this gene for insulin. We also use the same restriction enzyme. So what have we done is we broken up the bacteria. So we centrifuge these bacteria, And what do we get? We get plasmids. We break up the bacteria and we get plasmids. And then we cut these plasmids as well with the restriction enzyme. And what do we get? We get open plasmids like this. So number one, we got restriction enzymes and we cut the chromosome and we got the gene for insulin. Then we used the same restriction enzymes to cut the little circles of plasmids which we got out of a bacterium, and so we destroyed some bacteria and we got those out of those bacteria. Now we have to mix the two. Now we've got to mix the two. Now when we mix the two, we get the brown this is the bacterial plasmid. This is the human insulin gene. But then it doesn't glue together. So we need an enzyme called ligase. And this ligase helps us to glue these two different DNAs. One is human DNA, one is bacterial DNA. The plasmid is made up of bacterial DNA. And the insulin is the human DNA. So now we have this What do we have now here? We have this, the human DNA, and then we have the bacterial DNA. And what is this called? This is called recombinant DNA. Recombinant DNA, why? Because it's a new combination of DNA. It's not one species, some of it is bacterial DNA and some of it is human DNA. And of course we have glued it together by this enzyme which has actually helped us to glue it together, which is called ligase. Now we take this recombinant DNA and we take another set of bacteria and then we mix all this up. Now hopefully we actually do electroporating. You do electric shock and these pores develop, and some of these plasmids may enter some of these bacteria. Now that's a very small percentage, 10 to 20%. So if you have 100 million bacteria, maybe 20 million contain these. Then we have to identify these bacteria which contain the insulin gene. How we identify, we'll just study that. Now this one doesn't have it. This one doesn't have it. This one doesn't have it. We don't need these bacteria. They're useless. What we all need is this one and this one and this one. So we collect all these. Then we put it in a fermenter. And we culture these bacteria. We add the food. And these bacteria produce insulin for us. So these ones which are the insulin containing ones, we are going to put them in a fermenter, culture them, add the food, add the nutrients, and these bacteria are going to grow. And each bacteria will have a copy of the recombinant DNA. And all these bacteria will produce insulin for us. Now we separate the bacteria and we separate the insulin. And we extract the insulin and give it to people who are diabetic so that they can inject themselves with insulin. Now the trouble is that, out of 100 million bacteria, only 20 million actually contain that insulin plasmid. But how do we identify this? We can't be looking for it in every cell. So what we do is actually when we introduce the insulin, the human insulin DNA, along with it we add another gene which is either an antibiotic resistance gene or a fluorescent marker gene. So in the plasmid, we've also added that. So then we will add the antibiotic and we will figure out, okay, if the antibiotic kills it, then of course it doesn't contain the insulin, but if it is resistant, then we know it has got the antibiotic resistant gene and it's got the insulin gene. So well, that bacteria we need. Or what we do is we actually take it from jellyfish, we take a fluorescent. Fluorescent means which can, of course, when you expose it to UV light, it glows, fluoresces. Fluorescent marker gene. So then the bacteria can be, you shine UV light on it and you can see the one which shines, just like you have these glow paints. You can have them on your shirts, you can have them on your caps. And in the dark, you have these caps which will glow. So I'll be able to identify, okay, well, this student is from the school because he's got this glow cap, wearing this glow paint on his cap. So I can identify that child in the dark. Just like when we went for Hajj. The group that we went with had a special abaya, and you could tell that, okay, these people were from our group because they're wearing a white abaya with a green lace around the edges. So how do we identify the insulin bacteria? Now that is one of the biggest dilemma that genetic engineers face. Now the last topic is potential advantages and dangers of genetic engineering. Now naturally the advantages are of course far and few, but no more animal products are used, we don't slaughter animals, we don't take out their pancreas, we don't extract the insulin out of that. No more disease transmission. You see, remember I told you that when we gave animal insulin, we were a bit scared because that could resulted in some diseases which were caused not by a pathogen, but by a prion protein, and that could cause diseases. We didn't know about that. Then there's no religious or ethical issues in genetic engineering. Bacteria is not haram or halal, anything. So no religious or ethical issues. Then commercially we can fulfill the supply and demand needs. It's just grow in fermenters, and whatever the need is, we can always increase the supply or decrease the supply as the need decreases. And of course we can produce it all the year round. All we need is a lab where we have these genetically engineered bacteria, and we just keep them there and they produce insulin for us. Now, dangers of genetic engineering are of course very, very frightening, and of course we have a lot of biological warfare movies on that. Now, the only fear that we have is, what if these bacteria escape? There's a lot of security in these labs where there's genetically engineered bacteria. So what if these bacteria escape and they reach waterworks, so they reach the soil, and then we have insulin in the soil, and we have insulin in the water courses, in the ponds, in the lakes, and the rivers. So we don't know what it's going to do to marine aquatic life. It's going to kill the fish. It's going to accumulate in plants. The fish that eat those plants, I would have too much insulin in them. What it could do to us if we eat those plants, or if we eat those fish. Then if these genetically engineered bacteria somehow enter the water supply of a city or a state or a country or anywhere, now people drinking that water will die in millions. Why? Because you drank water and that contains genetically engineered bacteria, and they start producing insulin in your body. You see, insulin is produced according to the needs of the body. Like for instance, you ate something sweet. You had a piece of chocolate cake. Well, a little bit of insulin is released. You had 2 pieces of chocolate cake. A little more insulin is released. But if you take insulin from the outside, if a normal person injects himself with insulin, he's going to kill himself. That's the way how you can commit suicide, is to inject yourself with insulin. So if we get all this insulin in the water, and somehow, I know it's a bit far-fetched, because we've got pepsin and we've got HCl in the stomach, and that should actually destroy the bacteria. Well, what if it doesn't destroy the bacteria, and if insulin starts being produced in your body? And insulin cannot be produced in your body. It has to be produced according to the need of the body. It should be only produced when your blood glucose level rises. And everybody can't, like for instance, if you're not a diabetic and you inject yourself with insulin, well, there goes your blood glucose. It'll just plummet down. It'll go to 0 or less, and you'll die. Because the brain needs glucose. So dangers of genetic engineering are the only is that if these bacteria escape, if they somehow reach the soil, or if they go into the water works, these bacteria are going to produce insulin for us in the water, in the soil. And of course, this is one of the biggest fears of genetic engineering, that what if these bacteria escape? Millions of people will die. Now, just a recap of another topic which I did in a previous chapter, but I just want to do it a little more profoundly in this video. And this is about natural selection. Natural selection is variation is present. Environment changes. Survival of the fittest. Better adapted survive. Pass on the advantageous alleles to their offsprings. Evolution occurs. So number one, variation is present. So let's look at giraffes. There were once upon a time small-necked giraffes, medium-necked giraffes, and long-necked giraffes. Now the environment changes, certain trees died away. Now the small-necked giraffes could not get their food because there were no small trees left. So the medium-necked giraffe survived and the long-necked giraffe survived. Now with the time, more environment changed. The medium-necked giraffes also did not survive, because the trees that were remaining were all very tall and only the long-necked giraffe could access those trees. So when there were only the long-necked giraffes left, then of course what happened? The long-necked giraffe passed on this advantageous allele of long neck to its offsprings. So now all we have is the long-necked giraffes, and we say evolution has occurred. We say evolution has occurred. So once upon a time there were small-necked and medium-necked and long-necked giraffes. So all three were present. Now only the long-necked giraffes are present. So there was variation. Environment changes. Certain habitats are destroyed. No longer those trees are present. Now in this changing environment, which one is better adapted? The medium neck is not adapted. It can't suddenly grow a long neck. Or it can't start changing its food and start eating something else and start eating smaller trees or whatever. So the better adapted survive and pass on their advantageous alleles. We don't say genes. Genes to hum sab mein hai. We all have the gene for blood group, but we have different alleles. And they will pass it on to their offsprings. And then we say evolution occurs. Now another example which I like to talk about is oak trees. Now you see what is the difference in them. They have different what? They have different root lengths. So have different alleles for root length. Well, this is just an example which I have come up with. You won't find it in any book. Now look what happens. There's drought. Drought means there's no rain for years or months. Now the ones with the short roots, these are at a disadvantage, so these will die. Over a period of maybe 10, 15, 20 years. Now the only ones left will be this and this. The medium length roots and the long length roots. Because there's been drought. And as the time proceeds, if the drought continues, then these will also die. And only the long root length will survive. So you understand now why I said variation is present and why I said variation is a must for natural selection and evolution. You see, if there wasn't variation, if there were only short root length trees, there was only one type. And of course, if the environment changed and there was drought, then bas khatam ho jate, these will all die. But what I said is the first ingredient for evolution to occur is variation must be present. Then there is an environmental change. Then those better adapted survive. pass on the advantageous alleles to their offsprings, and evolution occurs. So I've given you two examples. One is of the giraffes and one is of the different trees which have different root lengths, to make you understand what is natural selection and evolution. So this finishes the entire 5090 O-level biology, and I wish you all the luck. And please go through these videos, pause them, make your own notes, and then please do all the exam papers on this chapter and see if you can answer the questions or not. And thank you very much.